Chapter 7, Section 1. Life is cellular. Look closely at a part of a living thing, and what do you see? Hold a blade of grass up against the light, and you see tiny lines running the length of the blade. Examine the tip of your finger, and you see the ridges and valleys that make up fingerprints. Place an insect under a microscope and you see the intricate structures of its wings and the spikes and bristles that protect its body. As interesting as these close-up views may be, however, they're only the beginning of the story. Look closer and deeper with a more powerful microscope and you'll see that there is a common structure that make up every living thing, the cell. The discovery of the cell. Seeing is believing, an old saying goes. It would be hard to find a better example of this than the discovery of the cell. Without the instruments to make them visible, cells remain out of sight and therefore out of mind for most of human history. All of this changed with a dramatic advance in technology, the invention of the microscope. Early microscopes. It was not until the mid-1600s that scientists began to use microscopes to observe living things. In 1665, Englishman Robert Hooke used an early compound microscope to look at a thin slice of cork, a plant material. Under the microscope, cork seemed to be made of thousands of tiny empty chambers. Hooke called these chambers cells because they reminded him of a monastery's tiny rooms, which were called cells. The term cell is used in biology today. Today we know that cells are not empty chambers, but contain living matter. One of Hooke's illustrations of cells is shown in Figure 7-1. In Holland, around the same time, Anton van Leeuwenhoek used a single-lens microscope to observe pond water and other things. To his amazement, the microscope revealed a fantastic world of tiny living organisms that seemed to be everywhere, even in very water he and his neighbors drank. Cell theory. Soon, numerous observations made it clear that cells were the basic units of life. In 1838, German botanist Matthias Schilling concluded that all plants were made of cells like the one in Figure 7-2. The next year, German biologist Theodor Schwann stated that all animals were made of cells. In 1855, the German physician Rudolf Virchow concluded that new cells could be produced only from the division of existing cells. These discoveries, confirmed by other biologists, are summarized in the cell theory, a fundamental concept of biology. The cell theory states, all living things are composed of cells. Cells are the basic units of structure and function in living things. New cells are produced from existing cells. Exploring the cell. Following in the footsteps of Hooke, Virchow, and others, modern biologists still use microscopes to explore the cell. However, today's researchers use microscopes and techniques more powerful than the pioneers of biology could have ever imagined. Researchers can use fluorescent labels and light microscopes to follow molecules moving through the cell. Confocal light microscopy, which scans cells with a faster beam or laser beam, makes it possible to build three-dimensional images of cells and their parts. High-resolution video technology makes it easy to produce movies of cells as they grow, divide, and develop. These new technologies make it possible for researchers to study the structure and movement of living cells in greater detail. Unfortunately, light itself limits the detail or resolution of images that can be made with the light microscope. Like all forms of radiation, light waves are diffracted or scattered as they pass through matter, making it impossible to visualize tiny structures such as proteins and viruses with light microscopes. Electron microscopes. By contrast, electron microscopes are capable of revealing details as much as 1,000 times smaller than those visible in light microscopes because the wavelengths of electrons are much shorter than those of light. Transmission electron microscopes make it possible to explore cell structures and large protein molecules. Because beams of electrons can only pass through thin sample cells, and tissues must be cut first into ultra-thin slices before they can be examined under microscopes. With scanning electron microscopes, a pencil-like beam of electrons is scanned over the surface of a specimen. For scanning electron microscope images, specimens do not have to be cut into thin slices to be visualized. The scanning electron microscope produces stunning three-dimensional images of cells. Because electrons are easily scattered by molecules in the air, samples examined in both types of electron microscopes must be placed in a vacuum in order to be studied. As a result, researchers chemically preserve their samples with first and then carefully remove all of the water before placing them in the microscope. This means that electron microscopes can be used to visualize only non-living preserved cells and tissues. Scanning probe microscopes. In the 1990s, researchers perfected a new class of microscopes that produced images by tracing the surfaces of samples with a fine probe. These scanning probe microscopes have revolutionized the study of surfaces and made it possible to observe single atoms. Unlike electron microscopes, scanning probe microscopes can operate in ordinary air and can even show samples in solution. Researchers have already used scanning probe microscopes to image DNA and protein molecules, as well as a number of important biological structures. Prokaryotes and eukaryotes. Cells come in a great variety of shapes and an amazing range of sizes. Although typical cells range from 5 to 50 micrometers in diameter, the tiniest mycoplasma bacteria are only 0.2 micrometers across, so small that they are difficult to see under even the best light microscopes. In contrast, the giant amoeba, chaos may be 1,000 micrometers in diameter, large enough to be seen with the unaided eye as a tiny speck in pond water. Despite their differences, all cells have two characteristics in common. They are surrounded by a barrier called a cell membrane, and at some point in their lives, they contain the molecule that carries biological information, DNA. Cells fall into two broad categories depending on whether they contain a nucleus. The nucleus, plural nuclei, is a larger membrane enclosed structure that contains the cell's genetic material in the form of DNA. The nucleus controls many of the cell's activities. Eukaryotes are cells that contain nuclei. Prokaryotes are cells that do not contain nuclei. Both words derive from the Greek word karyon, meaning kernel or nucleus, and EU meaning true, or pro meaning before. These words reflect the idea that prokaryote cells evolved before nuclei developed. Prokaryotes. Prokaryotic cells are generally smaller and simpler than eukaryotic cells. Although there are many exceptions to this rule, prokaryotic cells have genetic material that is not contained in a nucleus. Some prokaryotes contain internal membranes, but prokaryotes are generally less complicated than eukaryotes. Despite their simplicity, prokaryotes carry out every activity associated with living things. They grow, reproduce, respond to the environment, and some can even move by gliding along surfaces or swimming through liquids. The organisms we call bacteria are prokaryotes. Eukaryotes. Eukaryotic cells are generally larger and more complex than prokaryotic cells. As you can see in Figure 7-4, eukaryotic cells generally contain dozens of structures and internal membranes, and many are highly specialized. Eukaryotic cells contain a nucleus in which their genetic material is separated from the rest of the cell. Eukaryotes display great variety. Some eukaryotes live solitary lives as unicellular organisms. Others form large multicellular organisms, such as plants, animals, fungi, and protists.